Good afternoon, everyone, and welcome to MitoAction's monthly expert series. Today, Dr. William Copeland, Chief of the Genome Integrity and Structural Biology Laboratory and Principal Investigator of the Mitochondrial DNA Replication Group at the National Institute of Environmental Health Sciences, will join us to discuss understanding diseases of mitochondrial DNA maintenance. My name is Stephanie Harry. I am one of the patient support coordinators at MitoAction and will be your host for today. Today's presentation will be recorded and available on MitoAction's website in the coming days, as well as on our podcasts, Apple Podcasts, Google Play, and Spotify. We encourage you to ask questions throughout the presentation using the Q&A feature at the bottom menu bar of your screen. If you're calling in via phone, please feel free to submit your questions to us by email at info@mitoaction.org. We will do our best to get through as many questions as possible by the end of today's presentation. Understanding the process of mitochondrial DNA replication is very important as we engage in conversations about mitochondrial disease. Over the last years, I've had the privilege of working alongside patients and families affected by POLG, POLG2, and TWNK genes. This presentation will focus on the diseases that disrupt the normal process of copying our mitochondrial DNA. We're so very honored to have Dr. Copeland with us today to summarize the genes that are involved in copying our mitochondrial DNA, explain how they participate in preventing or causing mutations, and share more about the consequences of these mutations on mitochondrial function and health. Dr. William Copeland received his PhD in biochemistry from the University of Texas at Austin in 1988. He then completed his postdoctoral training at Stanford University of Medicine Department of Pathology with Dr. Teresa Wang, studying the human DNA polymerase alpha primase complex. In 1993, he joined the National Institute of Environmental Health Sciences in Research Triangle Park in North Carolina and is currently the head of the Mitochondrial DNA Replication Group in the Genome Integrity and Structural Biology Laboratory within the Intramural Research Division. He's also the Chief of Genome Integrity and Structural Biology Department at the NIEHS. Dr. Copeland has previously served as president of the Mitochondrial Research Society and as co-chair and chair of UMDF Grants Committee, Scientific and Medical Advisory Board, and Scientific Symposium Planning Committee. Dr. Copeland's group studies the molecular effects of disease mutations of DNA polymerase gamma, is its accessory subunit in the mitochondrial DNA helicase. He has more than 30 years of experience in research of mitochondrial DNA replication and has pioneered the characterization of the human DNA polymerase complex. He was the first to clone and characterize the genes for the mitochondrial DNA polymerase, POLG, and the accessory subunit of this polymerase, POLG2. The current projects address the role of human polymerase gamma and mtDNA mutagenesis, study of the molecular effects of disease mutations in the polymerase gamma, and are elucidating the role of human polymerase gamma and induced mitochondrial toxicity caused by anti-HIV nucleoside analogs. Dr. Copeland, we are so excited for this presentation. Thank you so much for joining us today. Thank you so much for all the research that you do for our community, and just, you're already getting hearts. So people just love you. We really, really appreciate all of your hard work. So without further ado, I'll go ahead and turn it over to you and let you share your screen.
Great. Thank you, Stephanie. Thank you for that most flattering introduction. So if I can get this right. Get a little pointer going. So as Stephanie alluded to, I'm at the NIEHS, and we're focused on environmental health sciences. We are one of the NIH institutes, but we are not in Bethesda. We're in Research Triangle Park in Durham, North Carolina. The Research Triangle is called the Triangle because the 3 points are the 3 universities here: UNC Chapel Hill, which you can see here, some of the medical school. Duke University is the other point, and NC State. As Stephanie alluded, I'm heading up this Genome Integrity Laboratory, which is a department. And we're really fortunate in the Research Triangle Park because we have such, not just in our department, but within Duke and UNC, we have such incredible expertise in DNA replication and DNA repair. We were the recipients of 2 Nobel laureates in 1995, to Paul Modric at Duke and Aziz Hankar at UNC for studying DNA repair capabilities. So enough with that. So today, my group studies, I've been focused for the last 30 years on mitochondrial DNA replication. And so what I want to talk to you today is sort of outlined here. I know some of this may be kind of basic, but it just kind of bear with me. I want to introduce, the function of mitochondrial DNA, how it is copied. We also call how it is replicated, introducing the four main players, Paul G2, Twinkle, encoded by the gene Twink, and single strand DNA binding protein. So I'll just describe how DNA is copied, how mutations arise through that process in a normal process, and how genetic diseases that affect these genes can aggravate this mitochondrial DNA replication and introduce another layer of mutations in the mitochondrial genome. And that's kind of this mitochondrial DNA mutagenesis and these Paul gamma related diseases. And then I want to end on a positive note, talking about some really new exciting therapies on the horizon that I've been trying to follow and participate in. And I think that's the future looks very bright right now. So again, our mitochondria are these little jelly beans. They're actually some can be very tubular actually in the cytoplasmic of the cell. And they have their own mitochondrial DNA. This DNA is circular, which is a lot different than our nuclear DNA. It codes for about a 16,569 base pairs, meaning you've got your bases A to T and G to C bases in this genome. It is a rather compact genome. It differs from the nuclear DNA, which you probably heard in the nuclear DNA has a lot of like junk DNA. Mitochondria doesn't have that, so there's not a lot of spaces for errors per se. We know it's maternally inherited. This is what our human mitochondrial DNA looks like if you spread it out on mica, and then you can visualize that with this process called atomic force microscopy. All right. One interesting thing about mitochondrial DNA, of course, is that it is you don't have one copy per cell, but you actually have thousands of copies per cell, and that we call copy number. And that copy number varies greatly from tissue to tissue. Some tissues like heart and skeletal muscle contain a lot, where we know blood cells don't have hardly any mitochondrial DNA because they don't need it. How does this compare to the nuclear DNA? So as I alluded, even though there's thousands, so like in a muscle cell, there's 3,000 copies per cell. Even though that is high compared to the nuclear genome, because this is 3 billion bases per copy, and there's 2 copies, so there's actually 6 billion bases in our nuclear genome. Compared to that, even though there's a high copy number of that, mitochondrial DNA only account for 0.3% of the total DNA in a cell. And that is a problematic technically when you're sequencing DNA. And I'll come back to that and some of the research I'll describe a little bit later. So the overall function, of course, of this mitochondrial DNA is to make the proteins. And so there's 13 protein genes on this, and that's these color boxes. And then there's also these small boxes, which are tRNA genes, and then there's ribosomal genes. So these tRNA genes and ribosomal genes help translate and the protein coding genes to make 13 polypeptides. The function of those 13 polypeptides are to then combine with nuclear encoded components that are then transported into the mitochondria to make up the electron transport chain. And this carries out the latter steps of energy production for the cell. So this whole process takes about 87 proteins. Only 13 of those, this is 6, 1, 3, and 2, are encoded by the mitochondrial DNA. They have to combine with their nuclear counterparts to form the complete complex to carry out that. And of course, the function of that is to make ATP. And of course, that is required to feed to all essential tissues require a large amount of ATP, and that cannot be afforded by glycolysis by just simple sugars, but they have to go through this oxidative phosphorylation to make abundant amounts of ATP. So the mitochondria we know make well over 90% of all the ATP in the cell. So how is DNA copied? So you've got, first of all, most of the gene products that function in the mitochondria actually come from the nuclear DNA. About 1,200 to 1,500 gene products are made in the cytoplasm, encoded by the nuclear DNA, then transported in to carry out the function. And again, some of those combined with the 13 polypeptides to carry out this oxidative phosphorylation. So this DNA, like most DNA, is copied by enzymes we call DNA polymerases. Okay, so DNA polymerase, basically all it does is help match up this adenine to thymine or cytosine to guanine on a growing, on an unwound template DNA. So here you have a DNA polymerase. The DNA is unwound by an enzyme called a helicase, and the polymerase will then extend a growing chain by adding the appropriate nucleotide across the template base. So this is called the primer or the growing chain, and this is the template. So in human cells, there are 17 human DNA polymerases. So you think, oh, that's a lot. What are they all doing? Well, I'm not going to bore you with this, but the only thing that's important for this talk is that there's really only one replicative DNA polymerase in the mitochondria. All these others, with the exception, some of these, there's a couple other enzymes that work in the mitochondria, but they don't function to replicate the DNA. So the only polymerase here that actually replicates DNA in the mitochondria is this Pol gamma. It's coded by the gene POLG, P-O-L-G. And but it doesn't work alone. So as Stephanie alluded, in the 90s, we cloned this. It's coded on chromosome 15. It's a large protein, 140 kilodaltons. That's one of the larger size, but not the largest. So this enzyme doesn't work alone, though. So this is a cartoon of a replication intermediate. Shown here, this is actually what we call the replication fork, where all the copying happens. This is a polyacrylamide gel of those isolated and purified proteins. And we need to purify and get very pure proteins to study the events going on. So let me introduce all the players here. So there's 4 essential elements that are required for copying the DNA. We have the Pol gamma, the Pol G, which is it's a large catalytic subunit. It contains both the DNA polymerase activity, and that's that actual copying mechanism, as well as an exonuclease activity. That's kind of a proofreading activity that's required to correct mistakes as it goes along. It doesn't work alone. It actually has a kind of a low affinity for DNA, but in the presence of the accessory protein POLG2, it allows the polymerase, the POLG, to stick to DNA much longer to carry out DNA. That's called processivity, the ability to extend long stretches of DNA. Otherwise, the polymerase would put on 100 nucleotides and fall off. And that we need a polymerase to actually go around this entire 16,000 bases. And with the POLG2, it does. DNA is unwound by this helicase called Twinkle. It was named Twinkle by Hans Spellbrink because when they did immunofluorescence staining of a cell, it looked like stars because they were basically lighting up the nucleoids in the cytoplasm, and so he called it Twinkle. That's the origin of that. This process, the way mitochondria replicates, it's a little different than a nuclear DNA, but it generates a long single-strand stretch, and that is protected and coded by this single-strand DNA binding protein. So these 4 proteins work together to carry it out. But the business of copying is done by the Pol gamma. So we also know this process can introduce mutations, and that kind of gets to the business of the disease things. And so in 1988, Doug Wallace discovered the first disease mutation leading to Leber's hereditary optical neuropathy. And now we know there's actually hundreds of point mutations and a large number of deletions that can give rise to symptoms and diseases, which all come under the umbrella of mitochondrial diseases. So mitochondrial can have mutations. And how does that work with that copy number? And that's kind of illustrated on this slide. So the mixture of wild type and mutant copies is a term we call heteroplasmy. And if you have a cell with 100% wild type, it's homoplasmic wild type. If it's 100% mutant, it's homoplasmic mutant. Rarely do you have that situation. Even in a normal person, you'll have some level of mutation. But the level of disease genomes, then that's what we call here in purple, is like this just genome probably has a deletion or a point mutation. The level of that is critical to whether or not a patient will get a disease. And when in the cell, when so many of certain percent of these genomes surpasses this what's called critical threshold, is when they will start getting the disease symptom. We know, and some of the we study a lot deletions. We know both homoplasmic or specific point mutations or deletions. That critical threshold is well above 50%. It's we're seeing somewhere even above 60% of those genomes in your cell. 60% or so have to be mutated with a disease-carrying mutation to give rise to the presentation of symptoms. And so, in other words, this large number of copies actually gives a mitochondrial of a huge advantage of buffering capacity as long as there's 50% or even 60% or 40% wild type copies, you're going to have enough to carry out normal function. Now, I'm not saying 60% mutated genomes is good for a marathon runner, but for most of us, that's probably okay or below that level. All right. But so where do these mutations come from? So I alluded they can naturally arise through the process of DNA replication. We also know mitochondrial DNA mutates, evolves faster than nuclear DNA, and that's from studying a lot of different organisms and through aging and different species. So I want to kind of go through what we know, and this is where our expertise is here. So the ability to copy good DNA is we call fidelity. So the fidelity of nuclear DNA replication is an order of 10 to the minus 10th. That means it's making less than about one mutation for every 10 billion nucleotides copied. That's pretty good, okay? And that's afforded by three events. We call this one, the DNA insertion fidelity. That's the actual by the DNA polymerase itself. You can think of this as compare this to typing or typing on a typewriter or a word processor. That insertion fidelity is equivalent to your overall typing. Now, the polymerase will make it's good about every 100,000 nucleotides will make a mistake. But then most of these enzymes have an exonuclease function within the same enzyme, and that's called this proofreading function. And that's equivalent, as you're typing, you make a mistake, you immediately hit the backspace and correct it as you go. And that's this proofreading function, okay? So then when you're finished typing your whole paper up or your whole page up, you then want to do spell check. And that's carried out in the nucleus by this process called mismatch repair. It's equivalent to spell check. It is a separate process, separate from DNA replication. It occurs after DNA replication. So this is for nuclear DNA polymerase. For the mitochondrial DNA polymerase, Paul gamma, is actually a very high fidelity enzyme. It makes about an error about less than 1 per million nucleotides. This proofreading function, which is intrinsic to the enzyme, accounts for about 100-fold to that overall process. However, you've probably heard that DNA mitochondrial lacks certain repair functions, and that is true. It lacks, there's several DNA repair processes that go on. I've just outlined 4 of the major ones. Mitochondrial lack a nucleotide excision repair. That's a type of repair required to take out large adducts from like UV damage, ultraviolet damage. It also lacks this mismatch repair. That's this process. And that's really important for the overall fidelity in the nucleus. All right, it does have this basic excision repair. That's really critical because that is what occurs during oxidation and oxidative damage. And we know that that's a lot of oxidation is going on in the mitochondrial. It also lacks ribonucleotide excision repair. That's really not a big player in overall mutagenesis. But this is sort of outlined here in this sort of cartoon of the processes we know going on in the nucleus. Only this basic excision repair is going on in the mitochondria. So because of that, We believe the high mutation rate is mostly due to the lack of mitochondrial mismatch repair, which means that the major driver of any mutation processes we see is from replication errors from the Paul gamma. So it's that reason why my group study heavily focuses on these players shown here, and trying to understand that process. So that I do need to emphasize mutations is a natural process, and we've studied this in aging and normal aging people, and it's very low. It is not going to contribute to aging or premature aging. It is not going to contribute to disease as long as these genes are all wild type. However, these genes, the PALG, PALG2, TWNK, and SSB, have all been identified as disease loci and by clinicians and by DNA sequencing methods, and they can give rise to these disease states. So the PALG, we know, can give rise to Alpers-Huttenlocher disease, progressive external ophthalmoplegia, mitochondrial DNA depletion syndromes as such. PALG2 can also give rise to this external ophthalmoplegia and a few other rather diseases. The SSB gene was only discovered 3 or 4 years ago. We were also participating in that research, and that causes this optic atrophy. And the twinkle also gives rise mostly to this progressive external ophthalmoplegia. Now, these 4 gene products are actually part of a larger list that we kind of keep track of what I call nuclear loci that identified again in the clinic that affect our mitochondrial DNA. And they come in sort of 3 flavors. You've got enzymes that work directly on the DNA to either carry out replication and repair. You've got, our favorites, PALG, PALG2, Twinkle, and SSB, as well as some of these other proteins were involved in overall repair pathways. Replication requires those nucleotide substrates, the free adenine triphosphate, guanine triphosphate, deoxyguanosine for those for the growing chain, and that is carried out by this nucleotide pool metabolism. And some of these diseases you may recognize, TK2 has been big in the news. And these are really horrific diseases because without the natural substrate, you can't even make DNA, and that gives rise to this depletion syndromes. Mitochondrial also can go through checkpoints where you can, through a fission and fusion process, that when there's too much damage, it can actually purge in a process called mitophagy. When that is aberrant by these gene products, you can get these sort of diseases, as shown here. So of these, PALG, unfortunately, is the big gorilla in the room, and that's because there are so many point mutations in the gene, up here's the gene, that have been identified. So I've got the gene up here. Here is the protein. And what I've shown here in this slide, these are the amino acid substitutions. So we pick one out. This is A467T. This is actually the most common. This is where an alanine amino acid is changed to a threonine. Actually, it's a rather subtle change, but it has a very drastic effect on the overall enzyme. They're kind of colored by their disease states. The ones in red cause that Alpers-Huttenlocher disease and other mitochondrial DNA depletion syndrome. It is a recessive disorder, but mostly affects young children. The rest of these are disorders that don't cause the DNA depletion. They cause deletions in your genome where a large chunk of the DNA is just missing. And that's afforded by these black ones are associated with ataxia neuropathy, and the majority of these are this progressive external ophthalmoplegia. And there's even some dominant ones here. So the majority of these are recessive, which means if you got one harboring just one of these and you've got a wild type PALG gene, you're fine. But with the exception of these cluster of dominant ones, so if a patient carries a dominant mutation, they will likely get a symptom sometime in their life. So unfortunately, Paul gamma is one of the most frequently inherited genetic defects in mitochondrial disorders. I think it accounts for upwards of 20-25% of all inherited mitochondrial disorders. Russ Senato and Bob Nabio have done some statistics and kind of estimated that approximately 2% of the population carries a recessive, mostly, pathogenic genetic variant. But the combined prevalence of both those recessive and dominant diseases, meaning that about 1 in 10,000 individuals will succumb to some level of Paul gamma mutations. Fortunately, most of those are late onset diseases, but not all. We know that the early childhood ones, the Alpers and the mild cerebral hepatopathy spectrum disorder, cause depletion, and those are very horrific situations for the patient. Later, if the presentation is later, it gives rise to these deletions, and it'll cause the neuropathy spectrum disorder or epilepsy or this progressive external ophthalmoplegia. Now, I do want to emphasize, just because what we know today, just because we've identified a person, a patient with known, let's say 2 copies of a known Paul G mutation, we cannot predict when. We can probably say, yes, they will get a Paul G disorder, but we don't know when. And that's kind of been illustrated in the literature. And so I guess I want to kind of go through an example of a review of a case studies of this T251I, that's threonine changed to an isoleucine. This is always found on the same allele with this proline changed to a leucine at this 587 position. All right, so this is usually found on one allele. And in this sort of cohort set, there's the patients also had these mutations on the other Paul gamma allele. Now, this is what I want to illustrate, is that we see here 3 sets of patients, all completely not related, that carry the same Paul G genetics. But you mean one allele has this this disease mutation pair, the other has this G458S. We've studied all 4 of these, or all 3 of these disease mutations. We know exactly what they're doing in the test tube. For example, the G848S is pretty much doesn't produce an active enzyme at all. This only has some partial activity. But look at the age of onset. This is crazy. This patient presented very early at 6 months with Alpers. But then you got 2 other cases, didn't present until 51 years of age with PEO and 73 years of age with ataxia neuropathy disorder. And so that's really baffling to us, is like, that means there's something else going on. Here's another case from Shimema Raman in London. She looked at 4 patients with this homozygous A467T mutation. So we had patient 1 presented at 3, 6, patient 2 at 6, patient 3 at 20, and patient 4 at 24 years of age. And 2 of those were still alive. And so the presentation, there's no formula given a certain Paul G genetics to say that this person is going to get it at this age and effect. So there's certainly other things going on that kind of can aggravate these Paul gamma diseases. One of those, of course, can be environmental factors. And this is something we can't stress enough, that if a family member or something is diagnosed with either a mitochondrial disease or especially Paul G, is to keep them as healthy as possible, keep them away from environmental factors that are known to are just very hard on the mitochondria in general. In addition, there's other mitochondrial DNA or nuclear DNA variants that we may not be known about that are participating and triggering the event along with the Paul G mutations earlier. So it's a very complex problem when you incorporate a mistake in the nucleus, and for example, a mutation in Paul G, to have to consider the rest of the nuclear DNA, the rest of those 3 billion base pairs, and all the environmental things that they're exposed to. So it's a very big problem. So the best thing is keep people as healthy as possible. All right, I want to turn to some focus that we've been looking at this disease, this progressive external ophthalmoplegia. This is a disease caused by both mutations in Paul G2, or the twinkle. And it was described in the early 90s as caused by deletions in a mitochondrial genome. So this gives rise to this inability to look right or left, which is called ophthalmal paralysis, or bleoptosis, which is the droopy eyelids. It's just basically the weakening of the external eye muscles. And what I understand, even compared to the heart, these external eye muscles use more ATP per mass of tissue than even the heart does. Because even in your sleep, you're always moving your eyes, you're always blinking all the day. So they're very energy utilizing tissues. And so the disease shows up there first, but it progresses to general muscle weakness, dementia, and many other symptoms. So this disease is characterized by these large deletions. So several years ago, we want to kind of look at these deletions to see, can we, actually sequence these and characterize these. And so we undertook a process of trying to sequence that and kind of ran into some problems. But after working it out, we developed a pipeline for looking at deletions, which we call lost arc. Because if you think of a circle and you miss a part, you're actually losing part of the arc of a circle. So that's where it comes from. Also, one of the authors, Scott Luhan, was a big fan of Raiders of the Lost Ark. So it's kind of was a play on words there. Anyway, this is a highly sensitive method, and it works by getting rid of the nuclear DNA. So what I alluded to earlier was that mitochondria only account for 0.3% of the total DNA. That presents a problem when you're sequencing. That means 99.3% of the sequencing reads goes to the nuclear. And we don't want that. We don't care about the nuclear DNA. That, to us, that is our garbage. So we get rid of it by digesting it away. Nuclear DNA is linear and mitochondrial DNA is circular. So we take advantage of enzymes that will chew linear pieces of DNA away. When we do that, we can enrich for the mitochondrial DNA over 1,000,000-fold. And from that, we can see a lot more of the mitochondrial DNA. And so we published this study of looking at about 20 individuals, about 22 patients with PEO, and 19 age-matched controls ranging from 17 to 93 years of age. And with that, we identified 35 million mitochondrial DNA deletions in these patients, of which about 470,000 were unique. So this is a lot. There's a lot of deletions. Before our publication in the literature, there was probably a description of maybe a couple thousand deletions, and now we've just kind of flooded the field. Unfortunately, we have too many. Let me kind of describe how this works. So this sort of illustrates the sensitivity. This is a tumor cell line. We've kind of worked out some of the background problems with that. This is the mitochondrial genome from 0 to 16 kb. And these black dots indicates the size of the deletion. And this is the sensitivity, where we can see down to 1 in 1,000,000. Now, why that matters is that if you send your DNA to a sequencing facility, they can see deletions at the 1% level. Remember I said there's about 1,000 or several thousand copies per nuclear per cell? Unless your deletion is above that level, they can't see it unless they amplify that specific deletion with this polymerase chain reaction, PCR. So we call this background. This is what our youngest patient, our youngest wild type patient looked like. And they had about an order of magnitude larger number of deletions, very low level of deletions. I need to stress, this is a low level, and will never get a Paul G disease throughout their lifetime. This is what one of our patients looked like. It was a 45-year-old with this homozygous AFRIC7T mutation. And they had a slew of deletions well above that background. And usually about 8K, there's about 8 KB size deletions and such. What that meant, though, the totality of these, most of these were rare, though, but the totality of that meant that 70% of their mitochondrial genomes carried a deletion. The majority of those were well below this level threshold. So they really weren't detected unless they did a specific amplification technique. And so we now have a process to look at very, very sensitive methods to look at these deletions. We illustrate this with what's called an arc map. This is our mitochondrial DNA. These lines here represent the deletion. So this is what in the literature you might have seen something called the common deletion. This is the common deletion here. It takes out about 4,977 bases. And so what this means is that this area here is actually deleted from that genome. Now, when compared to a point mutation, when you take out this much DNA out of a genome, it pretty much renders it useless. But there's no way for a post-mitotic cell to get rid of a useless genome. So they just stick around as long as they have an origin of replication. So we've colored these by the size as well as the frequency. So some of these deletions here approach that 1% level in this data set, and as well as what the ends of those pieces of DNA look like. So in this particular patient, 79% of the genomes contained a deletion, and that's why they have a disease. In that, and we're using a 25 milligram muscle biopsy. That's where the source of this DNA reads. So in that sample, we identified 26,000 unique deletions that have never been described in the literature. So there's just a lot. There's a lot going on. This is another one. This patient had this genome, this W748S, this with on one allele, and this arginine 1096 to cysteine on the other allele. And here we identified 24,000 unique deletions. Of a total of about 3 million deletions we detected. And again, 74% of their genomes contain a deletion. So we can look at this. We can also look at the patterns of those deletions. And when we looked at across our 20, we had 20 normals and about 20 patients, in an unbiased manner, the patterns of the wild type separate from those of the disease cases. And this is called ablation, talks about the kind of the degree of DNA that's actually deleted. So by looking at these patterns, and this ablation is actually a diagnostic marker of whether somebody's has this disease or not. And so we'd like to get this into a clinic. It's going to take a while. It's not a cheap assay to do, but we're getting there. It's certainly the cost of sequences has come down quite a bit. All right. I want to turn now to some more fun stuff in terms of what does the future hold for these diseases, and I'm going to focus on the Paul gamma related disorders. So what I've seen in the last 20 years, 25 years is that what's available to the clinicians and the medical doctors is there's just a lot more literature available for the clinicians, the families, and patients to help digest and to help, especially to help the clinician to make that correct diagnosis. What will really also help that clinician to make the correct diagnosis is a molecular analysis of the DNA, and that's doing this whole genome sequencing or whole exome genome sequencing to quickly identify not just Paul G, but any nuclear DNA mutation that can give rise to these diseases. You may be aware, we actually manage one data set, this Paul G mutation data set, where we started over 20 years ago to help kind of categorize and kind of just the Paul gamma, the papers associated with the Paul gamma diseases. And they're done so by the actual position of that. What's also afforded is that these are now mostly covered by health insurances, which is a great thing. And a lot of that is because the cost of sequencing has come way down. So you may be aware that let's compare to computer memory. When I first bought a computer, I think my computer had 256 kilobytes, and that was a big deal. And now you get a computer, it's got a half a terabyte, okay, and it's the same cost. So sequence, compared to computer memory has come down about 1,000-fold. But sequencing, DNA sequencing has come down by well over 1 million, probably 10 million fold. And so compared to what we could do 20 years ago, it would just take millions and millions of dollars to do one person. Now you can do a whole genome sequencing for $1,000 or a couple $1,000, and mostly it's now covered by health insurance, which is great. So we also, in terms of research, we have better disease models. to develop therapies. We have cell-based models, and these can be derived directly from patient cells per se. We also have some really good mouse models. We've been involved in generating a Paul G mouse. So there's Alexandra Trefanovic developed a Paul G mouse, and we developed a Paul G2 mouse. And Anu Sumalayan at Helsinki developed a Twinkle mouse. So there's mouse models for all of these, which is wonderful. There's also zebrafish models for both Paul G and Paul G2. And the development of organoids. And so let me just illustrate that. So there's now a zebrafish Paul G2 knockout. There's a Paul G knockout as well in zebrafish, and they're now being used to screen for drug treatments. And zebrafish are kind of a really, compared to a mouse model, you can do it much faster. It's a little bit trickier because they are actually fish. And so it's just a different way to study things. I've been following this work by this woman, Christina Lang, for a while, and she is now developing what's called organoids, and she's developing directly from Paul G patients. And so an organoid is where they take embryonic stem cells or stem cells from a patient and will differentiate those into in vitro organs, basically. And you can use those to then study a whole range of capacities, and I'll come back to that, of how that can be used. So the really good models now. What about therapies in the works? So I've kind of outlined here some of the known therapies. What's been used for a long time now is this antioxidant therapy, and that's been really critical because we know mitochondrial can generate a lot of oxidative damage, and oxidative damage can cause inflammation, and inflammation is one of the major drivers of a lot of the symptoms. So treating it with antioxidants is really critical. So there's drugs like CoQ10. There's drugs like, I know Children's Hospital of Philadelphia is running a NAC trial in acetylcysteine antioxidant therapy, and that's working really well. There's the Edison drug, EPI 743, which was an antioxidant type drug. That was useful for a number of reasons. Wasn't so useful for the early childhood, the Alpers cases, but certainly some other patients as well. I do want to emphasize, this is a therapy. It's not a cure, except gene therapy. None of these are really going to be a cure. But the antioxidant is sort of treating the consequence. It is not even treating the Paul G enzyme itself. It is treating the effects that the secondary mutations are causing. What I mean is by that is the mutations in the mitochondrial DNA. There's also a large number of anti-seizure and anti-epileptic drugs. Anti-seizure drugs were used quite a lot against some of the Alpers patients early, and some of those, especially valproic acid, actually showed a significant problem. But there's better ones now. For example, there's a new vitamin K analog. This is that's been developed to suppress seizures. This is in a zebrafish model of Paul gamma. This was developed by a ex-postdoc of mine, Serene Chan. She now has a company developing this drug into clinical trials. And so does not have all the side effects of things like valproic acid does. So that's very promising along the lines. Nucleotide therapy. Now, nucleotide therapy has been incredibly useful for those nucleotide metabolism diseases such as TK2. So you could treat TK2 patients with thymine or thymine monophosphate, and it will really reverse the effects of that, the defects nearly 100%. It's a little bit trickier with Paul G diseases, but it is shown effect on some of those. So there's two important papers that have looked at nucleoside supplements treating mitochondrial DNA depletion syndromes. Those aren't TK2 type. This is really dealing with Paul G type of diseases. And again, Christina Lang has used deoxynucleoribonucleoside treatments to rescue some of the Paul G mutations when they've been stressed with this lithium bromide, which helps deplete it to sort of like um, makes everything go faster. But she's shown that you can actually rescue that. Now, I do want to stress it really depends on the type of Paul G mutation. Some of these can be rescued by deoxynucleotides or nucleotide therapy. Others won't be. So it's really kind of a hit or miss, unfortunately. Um, Metformin, you've probably heard about, a very popular anti-diabetic drug. Again, Christina Lang has shown that metformin can ameliorate some of the defects with the Paul G mutations. So that's really promising and something to pay attention to in the field. Moving forward, there's a large number of efforts, both Stanford, both in Sweden, both here on the East Coast, where they're looking at drug screens. You're looking at these drug libraries and repurposing old drugs to see if they can work against mitochondrial or Paul gamma-related diseases. And I think that's how Christina Lang discovered the effects of that metformin can help. And of course, the cure-all is probably gene therapy. I don't, you've heard about CRISPR technology that does work. There's off-side effects that I think people are trying to work with. And then there's a lot of politics and ethical problems with that. So just pay attention to that. That's something probably more in the distant future. So with that, I think I'd like to stop and take questions. And should I unshare or?
Yes, go ahead and unshare.
Okay.
Um, Absolutely amazing. I love how you broke it down for us, and I really, really appreciate you kind of bringing to the community like, okay, focus on this. This is up and coming. This is like where the research is heading. That's really, it's really, really helpful to hear all that's going on with the research and where we're heading. We've definitely had some questions filter in and would like to take some time to try to tackle some of these. Um, one patient was curious if you could explain CPEO plus due to POLG1 mutations. And I know you talked about kind of like where some of the research is heading and stuff, but is there anything specific to CPEO plus and POLG1 mutations with what you were just talking about in regards to treatment?
Yeah, I may be speaking out of tune because I'm not a clinician, but I understand the plus is just the more advanced state where you've got the secondary or other symptoms. I mean, the classical symptom is just the eye disease, but what I wanted to stress is that it just starts there. And depending on, if you get this early, you're going to get other symptoms along the way. You're going to get general muscle weakness. You're going to get hopefully no more than that, but there's other problems because it is a systemic. You have defects in all the cells, and it's just presenting first in the eye muscles, but it will present later in other tissues.
So it's possible when you're diagnosed with CPEO plus, like if you haven't had your full genome tested, that you don't really know the cause of your CPEO plus, it sounds like.
Right. Unless you've got, unless they've done the sequencing, it could be. So it can also be sporadic, and that was meaning it may not have a nuclear gene defect at all. It may not have anything to do with Paul G or Paul G2 or Twinkle. It could just be sporadic accumulation of, you just got too many deletions in your mitochondrial genome that kind of replicated and out replicated the wild type copy. And so then it just out of a sporadic event, but probably
No,
more
that's
rare than, yeah.
Yeah, no, that's really helpful. And it kind of leads into the next question because there's a patient who was diagnosed with POLG in 2016, but the genetic testing had indicated POLG but didn't delineate between one or two. And
Oh,
they're
okay.
curious, like, how can I know which I have and does it matter? And so I think some of that probably touches base on how much technology has changed from 2016 until now. Can you talk about that a
Yeah.
little bit?
So yeah, I fight with the nomenclature all the time. So if it just says Paul G, it's also Paul G1. Paul G and Paul G1 are synonymous. They're the same. Um, and when so genes have a specific naming criteria. And when we discovered the human Paul G, we went to that. It's called Hugo, human name, human nomenclature for genes. and they had already reserved POLG for our gene. So, and we didn't know there was a POLG2. And so we went with that, POLG. And then it was discovered this accessory subunit, and so POLG2 made the most sense. So a lot of people call POLG, they also call it POLG1, they call it POLG-A. It's still POLG. Yeah.
Let's see. Oh, another question is on, okay. It says one patient asked on the point mutation slide, are all of those in exons or any of those in the
No, those are all in
introns?
exons. We do know yes, we find mutations in the introns as well. Um, unless they're close to the splice junction between the exons, they don't really manifest a problem. And what I mean by close, they've usually got to be within 4 to 5 nucleotides of the splice junction. And we have splice junction mutations that affect those. And I think I listed some of those, but they're harder to list on that type of slide. But yes, we see mutations all over in the introns. And they usually don't cause a problem.
Do you, so you talked a little bit about where the therapy is going. Do you offhand know of current trials that are occurring? I know there's plenty of international folks here too, and I know that's a large
Right.
question to ask. And I know that anyone that's on here that's interested in clinical trials, there is clinicaltrials.gov where you can kind of look and see what clinical trials are available right now. But off the top of your head, do
Yeah.
you know what
Not,
exists?
I see Julie posted something from the Paul G Foundation. She probably keeps track better than I do. I don't know of any specific for Paul G at that stage in clinical trials now. There are certainly a lot of other therapies, like I mentioned, Children's Hospitals running a therapy with their NAC. There's a lot of other general mitochondrial therapies that are in clinical trials. But when it comes for the Paul G type or these diseases, no, I'm not offhand.
Okay. And I know that they are doing, for LMEPRTide, they're doing an expanded access program for some patients with POLG. So that might be something to
Right,
investigate as well.
right,
Uh,
right.
Another patient said she wanted to thank you for emphasizing the significance of environmental factors with mitochondrial stressors on POLG. She said it's validating to hear an expert acknowledge that there's something more going on here when the same genetic mutation presents so differently. So I just kind of wanted to throw that out there to make sure you heard that. And
Let
then,
me,
yeah,
can I want to go a little further on that because I was kind of
yeah,
prepared for
go,
that one.
yeah, go
So
ahead.
this is a kind of a graph of damage to DNA as a function of time and the effect it has on oxphos capacity. Okay, so normally I mentioned we normally accumulate mutations in our DNA as a function of age, but for a normal person this doesn't matter. Everything with a mitochondrial disease is sort of accelerated. And so what these lines here mean is that's the age when they'll present with a disease. And of course, by tissue types, that will, some of the central nervous system will get it, and heart will develop much earlier. Now, see, in the presence of one of these triggers or environmental factors, what that does, we think, and these are just hypothetical graphs. What the mitochondrial toxins do is they basically push down or they put pressure on the mitochondria to perform even harder. And so for a diseased person, a patient, that may mean that their mutation rates goes higher. Or if for a normal person, it may not matter, but for a mitochondrial patient, that means they'll present much earlier and those symptoms are going to be much earlier. So it is really critical to keep as healthy as possible. For a normal person, maybe this doesn't matter, because we're well above the threshold of that tissue. But for a disease situation, it does matter. So, oh, I think we lost your voice, Stephanie. Oh,
You're right. I
oh,
muted myself because I didn't want to cough while you were
okay.
showing your slides. But if there's other slides like that, I think the slide was really, really helpful. If there's other ones that you felt like you had to skip because of time, please feel free to go ahead and share.
Okay. Okay. I can
Yeah.
send you those. Yeah.
Okay.
Yeah.
Uh, let's see. Um, just trying to sort through some of these questions. My niece is 7 months and her mutations are GLY848,
Right.
serpatogenesia, and
Pathogenic.
another, yeah, oh, okay, pathogenic. And there's
919.
another MET919, which means the last mutation is pathogenic. Oh, okay. So she's asking about contacting someone with in private. So I will follow up. I see a couple specific questions on the question log, and I can follow up with these patients and make sure that you get connected
Okay.
to the right people. If there are any other general questions, please go ahead and feel free to put them in the chat. I want to make sure that I get to them. The more specific questions, I can try to follow up with you guys when we are completed. If there are patients that are interested in somehow participating in research, like is there any way that the patient community can support you in research, offer their data? Is there anything like that? How do you interact with the patient community and that way with your research?
Mostly, well, like any patient samples we get, it has to be a collaboration with a clinician. So we've worked with, we work with people like Bruce Cohen, Russ Senato, Bob Navio in the States, and Rob Taylor and some of the European colleagues to get samples over there. But a lot of that, to be honest, is just working with patient groups like yourself, like the UMDF. And I can't stress enough how important that is, because we like to do our own research, but a lot of times without a purpose, it doesn't mean much. And so giving us that sense of purpose is a huge motivation for us just to work harder and to get that answer. And so I can't stress that enough, that's really, I was trained as a classical chemist, biochemist, and it wasn't until I started working directly with patient groups and such where I started really loving what I do. It's just, I can't emphasize that enough. So thank
Well,
you
and
for everything, the support.
I mean, it's really helpful. I think one of the, and we were talking about this a little bit earlier, I think one of the complications in regards to mitochondrial disease is there's, you can't have a treatment until you know what the issue is or the cause
Right,
is. And so like
right.
understanding how, Polgy, Twink, how all of those things work together in order
Right.
to replicate DNA, I mean, that's huge, and that creates future therapies and strategies for therapies. So, your research is so very important. Um, there's a question that came through that was asking about all of our family members carry one POLG mutation. Our youngest child recently died of Alpers since he carried two mutations. What is the chance that any of our family members carrying one mutation will show symptoms in the future? It
It won't.
depends. It
Yeah.
won't.
Well, Alpers is a totally recessive, meaning yes, they're caused by pathogenic mutations in Paul-G, but they're known to be recessive. So a carrier with one of those is asymptomatic. They shouldn't get an effect. It's still a good idea to not stress your mitochondria or not stay as healthy as possible, but everything the literature is showing is that they should be fine. One copy is sufficient. One good copy of mitochondrial of Paul gamma is sufficient, unless it's one of those dominant ones. But
Okay.
Alpers is not caused usually by dominant Paul gene mutation. They're usually too recessive.
Okay. So it sounds
Yeah.
important too, maybe for them to know what the mutations were for the
It is,
Alpers.
and the family genetics. I mean, you can just look at the parents and say, well, you're 30, 40 years old and you're fine. And so you'll probably will stay that way. But yeah,
There's a
family
new,
genetics are important. Yeah.
yeah. There's a new drug to treat Friedreich's ataxia. Do you think it would benefit Polg patients too? Are
Yeah,
you familiar
I'm
with that?
familiar somewhat with that. That I think that fits into that antioxidant therapy. I think if I'm thinking of the same one. And yes, that's all the antioxidants are really critical to help keep like the inflammation down. And that it's helping controlling the symptom and the consequences. It's not going to reverse the Paul gamma defects, but at least some of the downstream effects it'll help correct.
And I think that was really helpful, the slide that you presented where you talked about the antioxidant therapies and how, and just kind of made clear that like this doesn't treat the actual problem, but it helps
Right.
with that trickle down effect. Because sometimes as patients, it's hard to know, okay, well, why are we taking all these supplements and what are they doing or what's the purpose? But it's important to understand that it's not fixing the problem, but it's just
Mhm.
helping with some of the
Mhm.
symptomatic issues.
Right, right. Yeah,
So if
definitely.
there's one question today that you wish somebody had asked you, what would it have been and how would you answer it?
I'd say the future looks much brighter now. It looks really good. Somebody asked me, I think it was 10 or 15 years ago, what's my outlook? When do you see a drug coming on the market? And I was kind of like thrown back because I didn't see one. And it wasn't in the near future. And now there's just tons. There's so many different companies now that I know are working on different drug screens and therapies and such. So um, we're in a much better situation, and it's only going to get better. So just yeah,
So hang in there. I really appreciate you bringing this hope to our community today. And thank you so much, Dr. Copeland, for everything that you do and for just all of your research and for keeping us up to speed on where things are at. And know that if you guys have questions, further questions, please don't hesitate to email me at sharry, Harry, at mitoaction.org. And I can help with I'm one of the patient support coordinators here, so I can help with certain patient related things. Or if it's a question specifically for Dr. Copeland, I can get you in contact with him about that as well.
sure. Happy to help.
Thanks so much. And thanks everyone for attending today. And we look forward to seeing you next month.
All right. Well, thank you, Stephanie. Thank you.
Thanks. Bye.
Bye bye.